So thanks everyone for coming to Funga's seminar series. So I'm Colin Averill. I'm the founder and CEO of Funga. At Funga, we restore below ground fungal biodiversity to create outcomes in forests as a natural climate solution. And I am super privileged to introduce Dr. Liz Koziol today. She is a scientist, researcher, and I believe a type of professor at the University of Kansas. And she is also the owner of MYCOBLOOM, which is the only supplier of native old growth arbuscular mycorrhizal fungi in North America. Liz's work is truly an inspiration. I think it really is some of the most compelling evidence that old growth fungi do indeed exist. And if you can understand and harness those organisms, you can drive really exciting and meaningful restoration outcomes. When we talk about what we do at Funga all over the world, we often hold up Liz's work as an example of, one of the reasons we know this has so much potential. So really excited to have you, Liz. Over to you now. Excited to hear the latest and greatest about all the great work you do.
Sure. Well, thanks for that intro. Couldn't have said it better myself. And I think not only am I the only supplier of local ecotype fungi in the US, but I don't know that anyone else is really doing it in the world. So it's pretty groundbreaking stuff. How's that looking on your end?
It looks incredible. I love the spore heart.
Yeah, I like to take a lot of beautiful pictures of my fungi, and I'll show you some more. But so today I'm going to be talking mostly about the research I've done with native microbial applications in prairie, and then I'm going to dip a little bit into some of my more recent work in organic ag. But for those of you not from the Midwest, I thought I'd first describe the prairie ecosystem a little bit. As you can see, they're gorgeous. They're comprised of a major diversity of so many different kinds of plant species. And they're some of the most diverse ecosystems on Earth, but besides being extremely beautiful. So over a 1 meter step in some of the plots, these remnant prairies that I visited, you might step over 27 different plant species. So on a local scale, they're incredibly diverse. And so this is a historic range of the prairie, which ranged all the way down from Texas, where I think Funga is located, right? Are you Texas? All the way up to Canada. So most of the Central Plains was historically this tall grass prairie or mixed grass prairie community. However, today less than 4% remains intact in this ecosystem type. And near where I'm from, in the Chicagoland area, here's the example of Illinois. In black, in around 200 years ago, around 20% of that state, or 60%, excuse me, was in prairie. But with the advent of the steel plow, over time, this has been shifted out of this ecosystem type, and today less than 0.01% remains in this intact ecosystem. And that's because of the soil that these prairies made. These deep roots and all these microbes made this incredibly nutrient-rich soil, which is beautiful and black and excellent for growing things. And so with the advent of the steel plow, much of the landscape, if you've ever taken a road trip along I-70 in the Midwest, you'll see this. In the winter, it's brownfield, and in the summer, it's mostly corn or soybeans. So it's a massive shift of this ecosystem into agriculture. More towards the west, in that light green, a lot of this land is used for grazing. And both of these types of shifts in ecosystems can have massive effects on the prairie. In the example of the brownfield, right, the prairie's totally gone. None of those species exist. In the example of grazing, it's still a type of disturbance on this system because these lands are often overgrazed, which can lead to loss of particular species if they're particularly delicious to that cow. And so for the last 200 years, this landscape has undergone intense degradation, but it's also undergone restoration, especially in the most 2 decades or so, the last 20 years. And so when we're restoring these landscapes, often what is done, we go to a reference ecosystem, an old growth prairie, and collect all the seeds we can from that prairie. And then we put that into the landscape we're trying to restore. And so we've gotten pretty good at collecting seeds and harvesting seeds and getting some native plants to establish. However, there's a lot of different situations that occur along the way. So hopefully you can see my little pointer. But the top two are examples of what I would call a failed restoration. So in this case, it's mostly native weeds and some annuals that are native prairie, but short-lived species. And in other cases, it's a total failure. It's mostly non-natives that establish. And even though we can get some native species established, such as the case here and here, they're simply not as diverse as our seed mix that we're applying to that site, and they're nowhere near as diverse as our reference ecosystem. And diversity is super important because diverse systems, they're just simply better. They are better habitat for the insects, the plants, everybody using that space. They're more productive for the farmers that use that land for hay, for example. They're more stable and resistant to things such as drought and climate change. And they also help mitigate the effects of climate change better than less diverse systems, such as improving carbon sequestration, for example. So diversity is super important. So today I'm going to show you some steps to sort of try to bypass these in-between stages and get to this more diverse community. And I hope I convince you that the key to that is restoring not only the seeds and the native plants present, but also the microbiome. And so when I tell people I'm a mycologist, this is what they think my life is like, out there hunting mushrooms in the woods, which obviously I do. I think it's really fun. I love to play with mushrooms. But in fact, that's not how I spend my time at all. It mostly looks like this, playing at the microscope, because I work with a group of fungi called AMF, is their abbreviated nickname for arbuscular mycorrhizal fungi, which is a mouthful. So these organisms are microscopic. So yeah, I play a lot more at the microscope than in the woods. And so here is what these fungi look like. As I mentioned, they're subterranean, so they spend their entire life cycle below ground. They're not making those big, beautiful mushrooms. They're really just spores, which is this top figure. The spore is sort of like their reproductive unit. It's where their genetic information is stored in a very stable structure. And also what I target when I'm creating inocula. I want the fungi to be in the spore form because it's really stable. The other form they have is really the work of the fungi, which is their hyphal network. So down below is an example of this hyphae, which is sort of long filaments that branch out in all directions in the soil. And that's where the fungi do their work. They're out there interacting with that micronutrients of the soil. And so the plant and AMF symbiosis begins when the little seedling lands in the soil and its little rootlets begin to interact with that hyphal network. These fungi are an endomycorrhizal fungi, meaning they're actually growing inside of the roots of that plant. And it's there where they exchange nutrients. So the fungi use that network to collect things from the soil like phosphorus. And the plants use their leaves to collect carbon, which they then feed to the fungi. The fungi can't fix their own carbon. They have to be fed by that plant host. So in associating with these fungi, the plants can actually grow larger due to that increased access to all the soil nutrients that they can't reach themselves. And so the two main environments I study these fungi, on the left is the example of the prairie, and on the right, a brown field representing an agricultural space or a site that has been disturbed, pre-restoration site. And so what we know about the fungal communities in these spaces, the old growth fungi are more diverse. There's species that are endemic to these prairies that you don't see in other spaces, and they're really beneficial to the plants in that ecosystem. And these plants interact with those fungi for, in some cases, hundreds of years. These plant species can live up to, a human lifespan. Versus in agricultural soil, we have fungi that interact with plants for much shorter spans. So they don't necessarily have to be as beneficial, because even if they're not a good host, that plant's gone the next year. And so these fungi tend to be less beneficial, and the species that are there are fewer. And if you think about one reason that might be, for example, tillage can chop up those hyphal networks, which, literally chops those fungi into pieces, and many of them are degraded over time with repeated treatment like that. So we can look across the prairie and see how much these plants care about their native fungi using a pretty simple assay in the greenhouse or in the nursery. And we do this time and time again in the lab that I work in. And what we're measuring is, we call it the mycorrhizal response. And so up top, I just have a couple eggplants as example, where we grow it with prairie fungi versus without. And then we take that biomass and make a simple ratio of that, and that's the mycorrhizal response. So on this scale, if you're at a 1, you don't care about the fungi, you grow the same with and without. The example of this carrot here, this variety that's super sensitive fungi growing 10 times larger with than without the mycorrhizae, you'd have the mycorrhizal responsiveness of about 10. And so if we do this for a ton of plants from the prairie with their native fungi across the successional gradient, down here, the very early successional plants tend to represent the non-native plants in the community and some of the really early colonizers, if you think of like the black-eyed Susan. Versus on the right, our late successional plants are mostly plants that are only found in those endemic prairie communities, those old growth prairie systems. Here's what we see. So in this figure, every dot is an individual plant species. And so across the successional gradient, this data shows that the colonizing plants, the early successional and non-native plants, don't really care about the prairie fungi that much, in contrast to these late successional plants, which are strongly dependent on their native mycorrhizae. And so I have collected this database of all these different prairie plants. I think I have around 200 so far, and I do have that available if you're interested, and you can reach out to me after the talk and I can send that to you. But so let's just think about how this microbial filter will influence plant establishment. And I just picked 5 plants out of this database, and their mycorrhizal response is up there, up top. And so on the weedy end of the scale, black-eyed Susan has a mycorrhizal responsiveness of about 1. Versus on the far end, this late successional plant, rattlesnake master, has a mycorrhizal responsiveness of 10. So if we took all those 5 plants and we seeded them into a site for a restoration, what we would expect to see is this. Mostly these early successional plants that don't care about fungi are going to establish because, you know, they don't care about fungi. The plants that are strongly sensitive to the native fungi will not establish from that seed mix. And so if we transplant not only the seeds, but the fungi those plants care about, we would expect to see more of these plants that are strongly mycorrhizally responsive to that fungi to also establish. And so that's kind of the basis of my work and the theory in the lab group that I'm in at the University of Kansas, the Beaver Schultz lab group. And we've done this sort of microbial transplant, you know, dozens of times across this ecosystem now, and also beyond, and in other ecosystems such as organic ag. And we found it usually works really well to improve the diversity and reduce weed establishment in these sites. And so today I'm just going to pick out a few case studies from across this prairie range to talk about some of the methods and what we found specifically at those sites. But before I jump into that, I want to describe a little bit about how we've applied these microbes in restoration. People have thought of a ton of ways to put this stuff out there. And right away, I'm going to cross out some that I think are not very effective, and that's those at the bottom. So any sort of surface broadcasting or drenching of the inocula doesn't work really well, simply because of the ecology of these fungi. So they're subterranean microbes, and they're not adapted to be in the sun and to be desiccated. So blasting those spores on the soil surface, oftentimes they die. I haven't seen any good work with seed balls, although I could see it working. I'm not sure why it doesn't work, but in the few studies I've read, seed balls have not been an effective way to do it. And everyone is really excited about seed coating for these mycorrhizae. Unfortunately for microbes, they're huge. And so here is a picture of, I think it's an echinacea seed relative to a dime. And then I also put that same seed relative to the size of the spores of these fungi. So you can see a lot of these fungi are massive, even relative to this, you know, mid-sized seed. These spores are single cells, but you can see some of them with the naked eye. So they're actually quite large, and they're just simply too big to stick onto seeds. And so ways that we found to be effective to apply these microbes, I'll talk about several today, but all of the ones on top have in common some way of working that inocula below the soil surface. So I'll talk about one study where I did surface broadcast the inocula, but then I tilled it in. We've done that with sort of garden tiller and also a tractor implement. We've had some success with putting the inoculant in seed drill. And then we've also often applied the inocula via plugs, which I'll talk about a study using that first. And so I have some methods of how we put this inocula out available in a PDF. If you're interested in that, I can pass that along too. So the first case study I wanted to show was using that inoculated seedling or plug approach. We call this our nurse plant because it's nursing our baby microbes before we put it in the field. And essentially how this works is we take a plug, this is its little pot, these are pretty small little pots, and we put about 10% by volume of our inoculated on that seedling for 3 weeks in the greenhouse before we transplant it in the field. And this works great because that inocula has a plant host to feed it. Remember, these things can't fix their own carbon, so they really have to have plant roots to feed them carbon sugars so that they can grow. So this is an excellent way to introduce your microbes into a restoration. And so for this study, we had a couple different types of inocula we were experimenting with. And so the first was whole prairie soil. So for that, what we did is we went to a reference ecosystem. So that is an intact prairie that's never gone through plowing or anything like that. It's been a prairie since the last glaciation. And we collected a small amount of soil. And in that soil is everything a plant needs, including fungi, rhizobia, trichoderma. There's a nice, really fat worm in this particular scoop. So all the stuff that's in soil. From that same soil, we isolated just the native prairie fungi. So that was a second inoculation treatment. And then we had plugs with no microbes added. So we planted these plugs out. This is actually at 5 different restorations across the Midwest, and we followed something like 3,200 seedlings for this trial. We also seeded the restorations with 40 to 60 prairie species. So let's look at what happened to those seedlings the first year, those little inoculated nurse plants. In the black are the seedlings with just the fungi, and in the gray are the seedlings with the whole prairie soil. And for the most part, whole prairie soil and AMF perform the same. Both of these types of native microbes improve survival up top and leaf production down below. Considering what happened to that seed mix, I'm going to jump to year 3 of this restoration. Same, the dark fungi in black and the whole soil in gray. What we found is that the native fungi are actually slightly better at promoting plant community diversity than the whole soil. We're not totally sure why that is, but we think it has something to do with the pathogens that are probably present in that prairie soil. Down at the bottom, we also saw less invasive plant reestablishment in sites with that native AN fungi, probably also because the diversity is higher and there's more plants surviving and they're growing larger as well. And so this is some of the first data we'd gathered in comparing these 2 types of inocula, and it was a massive success, this set of studies. It showed that these native fungi, at least for grassland restoration, they're the most important microbe to include in a microbial mix, which is great because these things can be cultured in the lab. I sit at the microscope and do that. It does take a lot of time, but it's possible. And that's really important when we think about the remnants that we have for donor soil for microbial inoculation. That example of Illinois, there's so few pockets remaining of intact prairie we can use for donor soil. We simply can't scoop up enough to do restoration at scale. So this method shows us we can culture those microbes and it can still be an effective restoration tool. But speaking of scale, the next case study I want to talk about is how much of this mycorrhizae do we have to toss out into the prairie to see an effect, now that we know it's the most important microbe to add. And I like to call this study my handfuls to dump trucks study because I had this site. It was an old brome field that was tilled up. I applied a bunch of seeds, and then I applied anywhere from no inocula to handfuls of inocula per acre, around 2 to 7 pounds on the low end, all the way to literally dump truck loads per acre equivalent, which was the inocula was so thick you could see it on the soil surface. So that would be the representative of 8,000 pounds per acre, kg per hectare is pretty equivalent. So I'm gonna just briefly highlight what we saw early in the restoration. It's now in its 9th year, but the first year of the restoration, we did see some improvements in native biomass, but it took quite a bit. There was a lot of weed establishment that first year. If you look across the example plots, there's a ton of foxtail growing, which is a pretty common weed early in restoration. So about 512 pounds per acre was what was required year 1. Jumping forward to year 4, these restorations are coming along quite nicely. You can see me doing plant community sampling there. We found that native biomass was improved and non-native abundance was decreased around 128 pounds per acre. So I'd like to show you a little bit more of the data that I collected last year, the most recent data, which is awesome because it's one of the longest examples we have of inoculation treatments in this system for any lab group so far. So this is year 7 data. This is what these plots look like. They're really beautiful. They're very forb rich. So forb is a wild flower, a legume. You can see all the different colors patterning throughout this space and the diversity of plants establishing. So the first data I want to show you is that diversity. This is the floristic quality index, which is essentially how close a prairie is to that remnant, how many of those late seasonal plants we're finding. So on the X is our inoculation gradient, 0 all the way up to dump truck loads. And what we saw is the more native fungi you add, the better quality your prairie restoration is. There's about a 20% improvement with the highest densities of native prairie fungi. We dove deeper into what plants were driving that effect. We saw this awesome pattern of a trade-off. So the more prairie fungi we added, the fewer non-native plants were established, so native abundance declines. We also saw this improvement in late successional abundance and richness, that bottom right figure. So the more you had, the better your prairie was in terms of reducing weeds and having higher quality prairie species. And these are just some beautiful pics of these species. These prairie species are all just gorgeous. And so my figures are mostly in sciencey log transform scales, but I wanted to put some actual numbers on this line for you. And what we found with our highest inoculation treatments, we saw about 14 plants per 3-meter transect of these late successional seedlings versus only 10 without the fungi. So an improvement of about 40%. Oops. So I want to shift gears now out of the prairie into agriculture. So since I moved to the University of Kansas, I've been working with the Land Institute, which is a perennial agricultural group, and also some local farmers to ask in this space, do my native prairie fungi from Kansas help agricultural plants that are growing in this space in this local soil? And so the first study I'm going to show you was a partnership with a farmer in Lawrence, Kansas, Juniper Hill Farms, where I inoculated 5 pepper varieties and 4 tomato varieties with my prairie fungi. And this was all done in a hoop house. So if you're not familiar with the hoop house, it's an outdoor sort of greenhouse made out of plastic that's temporary. Down at the base here, you can see this black stuff that's all weed mat, and under that is soil. And so it's agricultural soil, which meaning it undergoes regular tilling and compression. So you would expect to have a pretty junky fungal community. And so I asked if my fungi could help these crops in this junky soil by using a side dressing approach where I put about 2 tablespoons of my prairie fungi mix on these different crops. And so every week I would go into these hoop houses and collect all the pepper biomass from these different treatments and weigh it and then give it to the farmer who would sell it at the farmer's market, which was super fun. I'm not going to jump into all the details, but I just wanted to give the main findings, which was that for both tomatoes on the left and peppers on the right, the non-inoculated plots, which were the ones in black, had more biomass for the fruit for both crop types than the non-inoculated plots. So this is some of the first evidence we found that these prairie fungi can also help non-prairie plants in what used to be prairie soil. And so very quickly, I'm sure you're getting tired of graphs, but I just have a couple more I want to show. And this is the effect of specific fungi applications. And so in all the prairie work that I showed, we were inoculating with a diverse mixture of prairie fungi. However, not all prairie fungi are the same. So different fungal species have different effects on crop growth and macro and micronutrients. And so this is a study looking at chard. These blue bars are all individual fungal species. In black is a mixture of all those species, and in white is the non-inoculated. And so you can see these fungi range from inhibiting chard biomass, which is the case of Gygospora gigantea there in the center, to improving it more than 33% relative to the control. We also have repeated this specificity test with prairie plants and other crops too. I just wanted to highlight some of my recent work looking at medicinal plants down below. And left, this was an in-field trial with cannabis, and on the right was a greenhouse study with echinacea in field soil. And same thing, we added just one particular fungal species, which would be those blue bars, that mixture in black and a non-inoculated. And we can see these fungi perform differently in terms of how much they help plants express their cannabinoids in the case of cannabis, and caffeic acid in the case of echinacea. So fungal species matter quite a lot. And another thing with this data set I wanted to highlight is that there's no best fungus. Depending on your crop, depending on your soil, depending on your plant host, what fungi is going to perform the best really differs. So if we just look at these set of graphs, on the bottom right here, Rhizophagus regularis wasn't really helpful for echinacea. However, with chard, it improved chard growth about 26%. And so it really depends. But what we often see is that the diverse mixture, which is the black bar, tends to perform as best as about the best fungus in these assays. And so a diverse mix is often your best bet. So after today, I hope I've convinced you a little bit that the key to successful restoration of prairie is thinking about these native old growth mycorrhizae and amending them back into this space. We also have some evidence that this can help organic agriculture. And these findings have been discovered across the globe, that these old growth soil inoculation treatments can improve establishment in many types of ecosystems, from shrublands to forests. So this data is really exciting, and I can't wait to see it as it emerges across the globe. So just a few more slides here. I just wanted to talk a little bit about MYCOBLOOM and why I started it. Was because after presenting this data for my dissertation over time, everyone was always like, how do we get these fungi? We believe you. We don't want weeds. We want late seasonal plants. We want diversity. Where can I get it? And having to say it's not available, or if you know a prairie that's going to be turned into an ag field, dig up that soil, it's brown gold. That was not very satisfying. So in 2016, I started MYCOBLOOM, which I think to date is still the only provider of local ecotype fungi for commercial use, although I think Funga is trying to do something specific to forest in their region. A little bit about MYCOBLOOM. I have 21 isolates in my collection of these AM fungi, more than a dozen species. And many of the fungi that I work with are from prairies, and they're not readily available in any other commercial mixes. All my fungi are from native landscapes, mostly from prairie. I do have custom mixes for ecotypic regions across that prairie gradient. My customers, so I started MYCOBLOOM as a tool for native land managers. However, it's expanded quite a bit. I have worked with so many people in these past years, from farmers to, cactus growers of all types. And so 85% of plant species associated with this fungi. So the potential applications are almost limitless. So with that, I'd just like to thank you and open it up for questions. And yeah, you can feel free to reach out to me via my email or link to my website, or find me on social media at MYCOBLOOM. So thanks.
All right, that was amazing. I'm gonna start pushing my Zoom reaction emojis right now. Everyone else should do the same. Thank you for that, Liz. So cool. It's cool to see the 7-year results. That's wild. It continues to work. I didn't realize you guys had replicated so many times in space too. That was really cool to see. I have like 2 questions, and then I'll open it up to everybody else. First, I have heard this great anecdote about where you find these fungi. And I know it's these old prairies, but Camille Delevaux also told me you guys sometimes go to old graveyards. Is that true? Can you share any more about that?
Yeah, especially where I'm closer to the eastern range, where I'm from in Indiana and in Illinois. That soil is so fertile, and it was so easy to plow. There were no trees that, if it was plowable, they plowed it all, And the few spaces they didn't plow tend to be things like hills with a bunch of rocks, or things that people saved, And one example of a place they stayed was cemeteries, right? So they would buy a small patch. These are really teeny tiny prairies, like 5 acres, that they hoped their whole farming community one day, you know, would use this space and live there, die there, and be buried there. But, you know, with the advent of the plow, fewer people were needed to live in these teeny tiny towns in the middle of nowhere, Illinois. So, you know, as people left, these spaces weren't claimed. And even the spaces that were claimed, they just dig, you know, whatever, a 6 by 6 foot hole when they're burying someone. And so for the most part, that space was undisturbed. And so, yeah, that's a great example of a remnant prairie where we've collected our fungi. We don't collect, you know, on top of the gravestones, so we try to go around to the edges.
Okay, that makes sense. That's so wild. I love telling that story, and I'm glad I've confirmed it from you.
Yeah.
Cool. The other thing I wanted to ask is, so earlier in the talk, you talked about measuring mycorrhizal growth response in the greenhouse, just to get a sense of who might be appropriate. And do you find that those mycorrhizal growth responses you see in the greenhouse are indicative of what, or predictive of what will happen in the field, or is it not that useful?
Yeah, I think it's useful. And so we've done some sort of, you know, inoculating in the greenhouse and also inoculating in the field with the same inocula. And it depends on the plant, of course, ecology, everything is context dependent, but essentially those plants that are insanely mycorrhizally responsive, you know, on the 20 end, you know, 10 to 20, those plants are really dependent on excellent fungi. And so the fungi that are in the field just simply won't cut it. And then inoculating with the prairie fungi, which are more beneficial for whatever reason, evolution, does really affect. And there's a strong correlation between greenhouse and field. Yeah.
Gotcha. So the extreme responders in the greenhouse are often responders in the field.
Yes, I would say the ones that are more, like some of the grasses are more facultative in their response, and in the greenhouse, they might not be super responsive, and in the field, they actually don't mind if there's junky fungi, some of them. Yeah, so it just depends on the plant, and a lot of that has to do with root architecture. So I don't know if anyone is a gardener here, but if you think about it, like an onion has very coarse roots and it doesn't have many fine roots. That's an example of a plant architecture type that is going to be really responsive to fungi. But our onions have been bred to, they don't mind if they're junky fungi. So they're not quite the same as our prairie plants, but you know, it has a lot to do with
Gotcha.
it.
That makes sense, and we take a similar approach. We do test a lot of our work in the greenhouse before we test it in the field, and we look for the tails of distribution in the greenhouse to really advance. So that makes sense. It's cool to see that you're taking a similar approach. All right, I'm going to open it up. Does anyone have a question they want to ask Liz? Feel
I
free to
have a
raise your
question.
Zoom hand. Oh, go ahead, Anne.
I raised my real hand. I hope that's okay.
That works. That allowed.
Awesome. Thanks. Hi, Liz. I'm Anne Marie. Thank you so much for sharing your data. This was really awesome. I'm curious to hear more about what a win looks like, both for you as a scientist, and then when you're thinking about the real world application for farmers, is that the same thing? So I can imagine for you, you might want a statistically significant difference, whereas for a farmer, any outcome that, you know, is higher biomass might be a win. So I'm just curious how you think about this and if these outcomes are ultimately aligned or what that looks like for you.
Yeah, I'm gonna break it down, I guess, my answer into native systems versus agricultural systems because they often have different goals, right? But so when I'm looking at my prairie and I see a win, it's because there's a seed in the seed mix. Like for example, lead plant is a plant that's in every remnant prairie across this range. However, in the seed mix, they often just don't grow. And so if I can see that plant species grow with inocula in the field, that's a win to me. So seeing these specific plants that are really sensitive, that are keystone species, which are often missing, reestablished, to me, that's a win because I care about diversity. But some land managers don't care about diversity at all. They just want some native plants there. So for them, what would be a win would be more, are there less non-native plants there? Because that's something they have to spend a lot of time and money to deal with. Land managers across the system spend about 25% of their effort throughout all the departments dealing with non-natives. So if we can limit non-natives for them, that's a win. And the best way to do that is to get these late successional natives to establish because they can live 100 years and there'll just be less spaces for these non-natives to reestablish. So those are the two kinds of major wins I think of for the prairie. For agriculture, it's all about biomass, right? They don't care much about anything besides, can I get more tomatoes? And so then it becomes more of a cost question. Can I get more tomatoes in this field and not spend, $12,000 per acre applying inocula? And so finding that minimum amount for them is the win. And that's something we haven't totally worked out in the agricultural system. I showed you that density thing in the prairie, but we don't know what's the minimum amount in the ag field. You need to apply it to see these effects. But I would guess, depending on the farmer, more specialty crop growers, seeing things like improvements in micronutrients and plant protein would be a win. But other farmers don't care about that at all. They just want as much corn as possible so that they can sell it and, feed our cows or whatever. So it depends on the grower too. Sorry, that was a long answer, but.
No, it was great. Thank you.
Here, I'll ask a question from the chat from Tony, who's asking if the isolation techniques for fungi vary between biomes or soil types. And I guess I would also follow on that one. Just what is your biggest rate limiter to scaling your inoculant production?
Yeah, it's heavy. That's the that's the issue. It's really, really heavy. But so in terms of isolating the fungi, our standard practice, we like to go when the fungi are in their dormant period. So in the spring and the summer, they're out there doing hyphae, growing, collecting nutrients. They're doing the work. And in the winter, they go into that spore form, which is their dormant form. And so we like to collect soil to acquire new inocula in the winter because we're targeting spores, because that's something we can collect at the microscope and then propagate for inoculation. And so that doesn't really matter much in terms of what type of biome. So to collect these spores, we essentially use a colander of different sizes. Like if you're thinking about catching your spaghetti, we have a big one to catch the big spaghetti and a smaller one to catch the smaller things. But our colanders are, on the scale of micrometers. So that can handle any soil type, the basic extraction technique. So it doesn't really differ between ecosystems. Even in a forest, if you're going to collect fungi, you would do the same thing. But so in terms of scale, yeah, that's a great question. And I'm struggling with that right now. MYCOBLOOM's been doing great, and I'm really having to grow very quickly, very fast. And so if you think about the smallest scale, we do these studies as these little pots. And when we started growing our fungi for cultures, we were growing them in pots this size, right? But that doesn't make sense when you're thinking about creating enough inocula to do acres and acres and acres. So our scale definitely has gotten bigger. Our pot size has gotten bigger. We need a lot of machines to mix this stuff up and also sterilize media. That's a major challenge. So we don't want to be passing pathogens around, for example, to different sites. So we have to start with a really clean media. And doing that at scale is a challenge. Yeah, the biggest piece of equipment we found so far to sterilize is about 2 cubic yards, which actually isn't a ton of media when you think about it. So.
Very cool. Yeah, I'll have to pick your brain more about scaling these inoculants, but I'll do it another time. Patrick asks if you have seen any really other ecosystem effects. He's really interested in riparian environments, so streamside. Does a restoration of these fungi affect water quality in any meaningful way?
Yeah, so I guess I haven't worked a ton in riparian spaces so far. I know a lot of wetland plants are not mycorrhizal or more facultative. So if it's straight wetland and it's an inundated system, these fungi don't necessarily like to be there. It's just too wet for them. However, we've done some sort of things that I think might answer this question. For example, on a new highway that was being built, the on-ramps kind of go up, and then there's this slope, this grade. And so we've done some inoculation trials along the grade where we have like little strips that have inocula and little strips that don't. And then at the bottom, we collect the runoff from these 2 strips. And these fungi do help with holding on to soil, both because they promote the plants, but then they themselves are made out of carbon, that hyphae network, which helps bind soil together and keep it stable. They also produce this protein called glomalin, which is really, really sticky, which helps soil stick together. So there are some effects on soil quality in terms of aggregate stability in different environments. And I would think that would translate to riparian areas. If it wasn't totally inundated, they could exist there and help hold on to soil in those environments too. Yeah.
That's awesome. Dr. Ian Ware.
Thanks, Liz. That was a great talk. I have lots of questions, but I think I'll hone in on kind of the pathogens. You've mentioned them a couple of times. You thought some of your patterns kind of emerging from whole soil inoculi were maybe potentially pathogen related. Obviously, the agricultural space is probably highly interested in pathogens as well. And then we think about them as well. We're seeing them pop up a lot in kind of these monoculture pine stands, with potential of having the application of, introducing mycorrhizae as a way to provide pathogen resistance. So I guess I'm just kind of thinking about, like, what are these systems that you're, restoring? Are these, these kind of degraded landscapes? Are they high in pathogens? Do you kind of have an understanding there? Obviously, you don't want to move them around. And I guess my second question is, like, how do you, I guess, put up, guardrails to make sure you're not spreading pathogens that way?
Yeah, I think it depends on the level of degradation in the system. So if we're thinking about like going from corn and soybeans to prairie, the overlap between plant species there is zero, right? Even the weeds that are in a cornfield are not going to be pretty much any of the prairie species. So in terms of specific plant pathogens, they're much lower when we're going from that space, from extremely disturbed to a prairie. We do some restorations that are more like a junky grassland. Like I showed that picture that was 100% big blue stem and 1% cicada husks. So we have done some inoculation in that space, which is the less disturbed but low diversity grassland. And we don't see huge effects of pathogens on these plants. It's not like they're melting away with, molds, but we do sometimes see for these plant species with the whole soil that there might be some specific pathogens. Yeah, and that's another reason whole soil might not be the best way to go, especially in, if you're thinking about for a business, it's illegal to transport soil across state lines in many places. So that's not even an option. But you can transport mycorrhizae, turns out, across the globe. There's almost no regulation. So yeah. But yeah, I think one other thought on that is with regard to, like, if you're having seedlings that you're growing in your little nursery that you are going to plant in the field, AMF do help prime plant defenses, even if it's, not something in that soil. So they can trigger jasmonic acid pathways, and for example, and then the plant is better defended when it goes in the field. And we have seen this effect, not for pathogens because they're kind of slow, but for herbivores. So if we have seedlings that are inoculated versus non-inoculated, put them in the field and they get hit with an herbivore, like, a little caterpillar, the ones that are inoculated have that defense system already going, and they're better able to defend than the non-inoculated seedlings. So.
Great, thank you.
There's a couple questions just in the chat, Liz, just asking for resources. I think people are super excited and would love any data you can share about, the graded strip and runoff example. So if you can, we're happy to share that somehow or point people to links. And apologies, I may mispronounce your name. I think your name is Mangi. Do you use any? Mangi asks, do you currently have any way to visualize where your projects are and show transparency using a map or something? And they're asking, like, maybe this could drive adoption of the MYCOBLOOM product, or generally, how do you reach out and have people discover what you do?
Yeah, I would say I'm a terrible business person, actually. I spend almost no effort on marketing outreach. I think I sent product samples to like 10 people ever in the course of this time. So it's very much a passive business, which, is what it is. I am who I am at this point. And so people often hear about MYCOBLOOM via my papers. So for example, the most recent contract I got, there was just a guy looking about how to limit weeds in a restoration. He's doing a huge project and found one of my papers about fungi limiting weeds. All my stuff is open access, so they read the science, and people tend to be inspired by the science first, which is awesome, I think. So that's, a lot of people have come to me from that. They're, sort of working professionals who are able to at least approach science, and that's how they find me. But then there's also word of mouth. I think that's very true. In terms of how to reach the farmers, that's even more challenging. They, tend to operate in clusters, and so you can get in with one farmer and then you're kind of in with the group of farmers, but how to reach them on a broad scale is a lot more challenging, and they're often working on shoestring budgets compared to some people doing restorations have a lot more money, because these things are showy and beautiful and in our public parks, for example, versus a farmer, even adding a few dollars per acre it's a lot. So
Gotcha. That makes sense. Well, good for you. It's just growing naturally. Tony asks if you've had any collaborations with people looking at native pollinators, and if there's anything you can share there.
Yeah, pollinators, no. But I have, so the first experiment I showed with the whole prairie soil versus just AMF versus nothing. We've done some cool experiments last summer. We just started it and it worked so well. We're doing it again where we did little pitfall traps. So we went into these plots and flush with the earth. We dug a little pit, put a cup down there, and then we're capturing the beetle and creepy crawly diversity in those plots. And so there are some effects. Basically, the plants with the more forbs, which tends to be the ones that have the more fungi, have the more diversity of these insects. So we've done that sort of thing. I don't know anything about pollinators, so I haven't reached out for a collaboration on that end. But we know forbs are the ones that the pollinators are attracted to these wildflowers. And so, yeah, I'm sure there would be some really easy to collect data there, but beyond my skill level.
Very cool. Yeah, it's cool just to see that you're triggering like a whole ecosystem-wide effect through the reintroduction of essentially keystone species, fungi. I think this is maybe a great last question. Ariel asks, how do you get involved? If people want to do more or engage more in mycology in general, what would you recommend?
Yeah, I often get a question like this from nursery owners who are super excited about, inocula, but don't really have the resources to buy MYCOBLOOM. And so I tell them, the things that I'm testing here, you could do. Like if you have a side nursery when you're just, growing little plants for your neighborhood garden club or whatever, you can use these techniques. You can find an area you think has good microbes and put little microbes on your seedlings and watch them flourish. So I think that is something that everyone could do in their own yard. That's really easy, just to think about the microbes. Now, I wouldn't go digging up your own yard and then inoculating your nursery and set it across the US because of the pathogens, but locally it's okay. And in terms of, I don't know, getting involved with MYCOBLOOM, great question. Spread the word. Yeah.
Awesome. We will tell everyone we know. Well, I think that about wraps it up. Thank you so much for sharing your work, Liz. I've been a fan for a very long time, and so it's cool to, just see you go deeper on it. I'm super impressed, and yeah, we take a lot of inspiration from what you do.
Yeah, well, thanks for having me. And for what it's worth, with the little pine seedlings, I heard that when they're young, they sometimes associate with AMF. So potentially something to think about.
Very cool. All right. Well, everyone should give Liz an emoji hand clap, and we'll call it. Thank you.
Guys.
And a real one.
Thank you all.
Thanks so much, Liz. Thank you everyone for joining.
Sure.
Have a good day.